This video corresponds to the worksheet that you will find on Canvas that allows you to review for chapters 18 through 20. In addition to going through this worksheet and the solutions here with me, you are also certainly encouraged to be going through the other worksheets for each individual chapter on Canvas, going through and reviewing your notes and making sure you're clear on all the terminology that we've presented in class, and going through the chapter for a source of additional review of these topics. You're also encouraged, rather than just watching through this video, to go through this worksheet, to go through it on your own first, and then use this as a way of making sure that you have the solutions correct. So our first question here deals with ranking the following molecules in order from the lowest boiling point to the highest boiling point. In order to answer this question, we have to think about what is going to drive the boiling points of molecules. The main thing that's going to drive the boiling points of different types of molecules is going to be intermolecular interactions. That is the attraction of one molecule in a container for other molecules in the container. And the greater those intermolecular attractions are, the higher the boiling point of the substance is going to be, and the higher the melting point of that substance as well. So thinking about intermolecular interactions, knowing that those are what are going to drive the boiling points and melting points of substances, we next need to look at what's going to increase intermolecular interactions, because we have to think about that in order to answer this question properly. And a list of things that will influence intermolecular interactions are going to be hydrogen bonding. If we have molecules that have NH or OH groups, those are going to be particularly capable of strong intermolecular interactions through that hydrogen bonding. Remember that hydrogen bonding is the strong intermolecular interaction, attraction between a negatively polarized oxygen atom and that very positively polarized hydrogen atom. We call that a hydrogen bond. It's not a covalent bond, but it's a very strong intermolecular attraction that we typically associate most with NH groups present in molecules or hydroxy groups, OH groups, present in molecules as well. So hydrogen bonding is going to be common in alcohol molecules, in amines, and things like that. We also, in addition to hydrogen bonds, looked at bond polarization as a factor that is going to influence intermolecular interactions. Because the more polar the bonds are, the stronger they are going to be engaging in intermolecular interactions. So if we have a bond between two carbon atoms, that's going to be a very nonpolar bond. If we have a bond between a carbon and an oxygen atom, that's going to be a more polar bond because the oxygen is more electronegative than carbon. And so that bond polarization leads to atoms in a molecule that are positively polarized and others that are negatively polarized. Since opposites attract, the bond polarization will lead to stronger intermolecular interactions between negatively polarized and positively polarized atoms. We can also look at, when thinking about intermolecular interactions, the carbon chain length. As the length of a carbon chain increases, so do the intermolecular interactions, because you have van der Waals attractive forces between those carbon atoms that are in the long carbon chain. So the longer the carbon chain, the greater the intermolecular interactions that we would expect. Those factors are generally less important than hydrogen bonding and bond polarization, unless we have comparison of a really, really long carbon chain versus a really short carbon chain, and also vary some of the polar groups in those molecules as well. Another thing that we can consider when we're talking about intermolecular interactions and carbon chain length is that if we decrease the number of branches in a carbon chain, that will generally increase the intermolecular interaction. So if we're comparing two groups that have approximately the same length of carbon chain, but one of those has some branches and the other one does not, the branches are going to result in decreased intermolecular interactions and hence decreased boiling point and melting point. So in other words, if you want to create the highest boiling point and highest melting point possible, you need carbon chains that are not branched. Branching is going to decrease the boiling point and decrease the melting point. So let's take a look at these molecules up top here and evaluate these factors to determine who's going to have the lowest boiling point as number 1 and the highest boiling point as number 5. We need to draw these out in order to visualize what each of them are going to look like so that we can evaluate these boiling points of the substances. So 3-methylheptane, we need to draw a 7-carbon chain because heptane is 7 carbons by definition. So go ahead and plug in our 7-carbon chain here. At position 3, we'll plug in our methyl branch there. We'll go ahead and draw next octane onto our list here. So octane is another hydrocarbon molecule, just carbons and hydrogen atoms in it. And it's going to have a grand total of 8 carbon atoms in its longest carbon chain. So we'll go ahead and draw that like so. No branches there. Then we draw N,N-dimethylpentanamine. So pentanamine indicates that we have a nitrogen atom directly bonded to a 5-carbon chain. So we go ahead and draw out our 5 carbons of that 5-carbon chain. And then N,N-dimethyl indicates that bonded to the nitrogen atom as well, we have 2 methyl groups. So we'll draw a methyl group here and methyl group here. And if you were drawing out the lone pairs, you don't have to for these line angle forms, but you would have a lone pair of electrons in that nitrogen so that it fills the octet rule and has no formal charge on it. Then we come down to N-ethylpentanamine. Just like the structure up top here, it's going to have a 5-carbon chain directly bonded to our nitrogen because that's the pentanamine part of this. So go ahead and draw that out as 5-carbon chain. And this one has just N-ethyl, so it's going to have an ethyl group bonded here. Now for this one, since we just have 2 carbons bonded to the nitrogen, in other words a secondary amine, we need a hydrogen atom in there to fill the octet of this, as well as we'll have that lone pair of electrons as well. So this is our N-ethylpentanamine. And then last on our list here is our 2-hexanol, our alcohol molecule recognized from the OL suffix there. So we plug in our 6 carbon atoms of the chain there. And at position 2, we're going to fill in our hydroxy group of 2-hexanol. So now, thinking about comparing and contrasting intermolecular interactions amongst these, so that we can go from lowest boiling point to highest boiling point, which of these is going to have the highest boiling point? So for come up with the highest boiling point, we're going to come up with what is going to have the greatest intermolecular interactions. Where if we rank these, we're going to have hydrogen bonding and bond polarization, the highest priority in determining intermolecular interactions, and carbon chain length and decreased number of branches, lower priority in determining how great the intermolecular interactions are. So of these molecules, the 3-methylheptane and the octane have no ability to hydrogen bond because they don't have any NH or OH groups, and they also don't have very polar bonds in there because they're just carbons and hydrogens, which have very similar electronegativities to one another. So to think about what's going to have the highest intermolecular interactions, that narrows it down to our 2 amine molecules, and our hexanol is the candidates for having the highest boiling point, the greatest intermolecular interactions. Thinking about these and evaluating the hydrogen bonding and bond polarization, hexanol with an oxygen atom in it rather than a nitrogen is going to have greater intermolecular interactions because an oxygen is more electronegative than nitrogen, so it's going to have stronger bond polarization because you have a greater difference in electronegativity between the carbon atom and the oxygen. It is also, like the nitrogen-containing molecules, capable of hydrogen bonding, and in fact, since the oxygen is more electronegative than nitrogen, the alcohol group forms an even stronger hydrogen bond than does the amine. So as a result, our highest boiling point molecule out of these is going to be the 2-hexanol. So I'm going to put that as number 5 here, because we're asked to rank from lowest to highest, with highest being number 5. Then, numbers 3 and 4 are going to be our amine molecules. Evaluating these amines, they each have the same number of carbon atoms. In one case, we have the NH bond present. That's our N-ethylpentanamine. In the other case, we have, in place of that hydrogen atom, a methyl group. So as a result, we're going to see that the N-ethylpentanamine is a more polar compound, and it's also more capable of hydrogen bonding because it has that hydrogen atom there directly bonded to the nitrogen. So it can act as both a hydrogen bond donor and a hydrogen bond acceptor. As a result, we're going to plug it in at number 4 on our list here of going from lowest boiling point to highest boiling point. So as the second highest boiling point of these, that leaves the N,N-dimethylpentanamine at position 3. Then we look at octane versus 3-methylheptane, and for those two, hydrogen bonding and bond polarization are very similar between the two, or non-existent is really a better way of describing that. So we look at the carbon chain length and the branching. The one with the more branches is going to have the lower of the 2 boiling points. So 3-methylheptane has more branches, so it's less able to engage in intermolecular interactions than is octane, which has just that straight chain and can pack really tightly. So therefore, listing these in order, we actually go from top to bottom, 1, 2, 3, 4, 5, to come up with the relative ranking of these from lowest boiling point at the top to highest boiling point at the bottom, driven by these intermolecular interactions. So now what we'll do is take a look at reactions of amines and amides. We'll start with the problem on top here. And what we need to keep in mind for this problem is that amine functional groups, which we see in our structure on the left, act as bases to react with acids. And certainly an acid example is hydrochloric acid, HCl. So as we think about what's happening here, we can think about the reaction mechanism, that is how the electrons have to migrate to take us from the reactants to the product. So on the reactant side, with our nitrogen atom of the amine acting as our base atom, the base is going to use its lone pair of electrons as a general rule to grab a proton from the acid. And we can show that by indicating that the arrow starts at the electrons on that nitrogen and finishes at the hydrogen atom. So we're showing a map of what's going on here. We're making a new bond between nitrogen and hydrogen. We illustrate that with our arrow. That forces the breakage of the bond between the hydrogen and the chlorine atom because the hydrogen can't exceed its allowed valence of having just one covalent bond. So filling in the products that result from this mechanism that we've drawn, this map of electron pushing arrows, we have our 4-carbon chain bonded to our nitrogen. Nitrogen is still bonded to 2 methyl groups there. And now it is newly bonded to a hydrogen atom that was donated from the acid. So I'm going to show that bond in red, since we showed our electron pushing arrow there in red, bonded to a hydrogen atom. A nitrogen that has 4 covalent bonds and no lone pairs of electrons will always have a positive 1 formal charge. So we'll go ahead and plug in +1 formal charge there as our product. Our other product of this reaction is going to be that chloride, which will have 4 sets of lone pair electrons because we broke that hydrogen-chlorine bond, and the electrons from that bond went on to the chlorine. The chlorine already had 3 sets of lone pairs to start with. It picked up a 4th, and that's going to result in it having a -1 formal charge if we use the formal charge calculation. Now to come up with the IUPAC name of the product that we've drawn here. And to come up with the IUPAC name, we need to recognize that our protonated amine is referred to as an ammonium ion. That's the type of ion that this is. When we have a positively charged nitrogen, we refer to it as an ammonium ion as the general type of functional group that is. To give the specific IUPAC name, we're going to incorporate that term ammonium into the name of this resulting molecule. In order to do that, we need to know what the name of the starting amine was, and we will change the amine suffix to ammonium in order to name that cation, in order to name that product of the reaction. So our name for our starting structure here, coming up with the name for that, we need to come up with the longest carbon chain beginning at the end closer to the nitrogen. So the longest carbon chain is going to be right here. We go with this as our longest carbon chain because this is the largest group that is bonded to the nitrogen. It's larger than those 2 methyl groups that are bonded to the nitrogen. So our 4-carbon chain with a nitrogen atom, nitrogen branch at carbon number 2, is called 2-butanamine. And then to indicate the other 2 groups that are directly bonded to the nitrogen, those shorter 2 chains, the 2 methyl groups, we plug those in as a prefix with N to denote that they're bonded to a nitrogen. So it's NN dimethyl for the 2 methyl groups there, the fact that they're identical, 2-butanamine. Now to name the IUPAC product of this reaction, what we're going to do is you take the name of the amine, and if the amine has become protonated, it's picked up that red hydrogen atom, then you change the name amine to the name ammonium. And that's going to designate to us that the nitrogen has a +1 formal charge. In other words, the nitrogen has reacted with an acid to become protonated. So let's go ahead and write out that name in the proper slot here. So it's going to be NN, we're not changing anything about the alkyl branches on this, NN dimethyl, and then 2-butan ammonium. And the ammonium is your indication there that the nitrogen needs to have a positive formal charge due to the fact that it has picked up that proton that we show in red. And then generally with these, we also name the anion that's bonded to it. And our anion here is our chlorine. A chlorine with a negative formal charge we refer to as a chloride, much like with sodium chloride. The chlorine has a negative formal charge. So NN-dimethyl-2-butaneammonium chloride is the full name for the product of this particular reaction. We'll come down to the next reaction here, which is an amine reacted with a carboxylic acid. Anytime you have an amine reacted with a carboxylic acid, the product of that is always going to be an amide. So the rule is here, amine plus carboxylic acid is going to give you an amide product. This was the type of reaction that we saw resulting in the formation of the peptide bonds in proteins. We took the amine group from one of the amino acids and reacted it with the carboxylic acid group of the other amino acid to give us an amide linkage or a peptide bond between two amino acids. So we'll do this here for our amine, which would be propanamine, and acetic acid, which you'll recall hopefully is the 2-carbon molecule that has our carboxylic acid functional group. So we'll go ahead and draw out the structure that results from this reaction, which is going to correspond to having the carboxylic acid carbonyl group intact, and then the OH group of that replaced with the nitrogen atom from our amines. So we'll go ahead and plug in a nitrogen atom here. We plug in the hydrogen atom that will have to be there in order for this to have no formal charge on the nitrogen, since the nitrogen wants to have that lone pair of electrons there in order to have no formal charge. So we get this as our major organic product, and keep in mind that if you're balancing the equation, the other product of this reaction is water, and you derive the water from the OH group here and one of the hydrogen atoms from your amine to balance out that equation. Now we look to coming up with the name of this molecule. And the way that we can name amides, such as what we have in our functional group here, is we can take the starting structure, the corresponding carboxylic acid, acetic acid, and we can change the name of that to have an amide suffix on it. So acetic acid can become acetamide. So that's our 2-carbon chain here. We will call our acetamide chain. So we're going to plug that in. Acetic acid became acetamide because now it has that nitrogen directly bonded to it. Another term of this, since acetic acid is synonymous with ethanoic acid, you could also call it ethanamide as well as a synonym of this. So that takes care of the part that we've highlighted in red and yellow here. And then we need to take care of the fact that we have our nitrogen atom bonded there. And so to do that, we need to name the alkyl group that's directly bonded to the nitrogen as a prefix here. And just like when we were naming amines and we described atoms that were bonded to the nitrogen atom as N groups, we're going to do the same thing here. So what we'll refer to that group that's bonded to the nitrogen as is an N group. So N dash, and as a 3-carbon chain with no branches, so N dash propyl acetamide or N-propyl ethanamide would be the complete name of this molecule. And if at the end of this molecule, the nitrogen, we just had NH2 and no alkyl group directly bonded, we would just call it acetamide. You would not need any alkyl name there with an N at the front. So in this case, though, we do have that 3-carbon chain there, so we need to name it as N-propyl acetamide as this structure. Okay. In this example problem, we look at the concept of proteins, in particular, protein denaturation. We ask, what is protein denaturation? Why is it important? And what are 3 ways that we can use to denature proteins? So this is something that we discussed that should be in your notes from our chapter on proteins, chapter 19. When we talk about protein denaturation, what we're referring to is the disruption of the 3-dimensional structure of the protein. Because in order for the protein to be functional, it is absolutely essential that it have the correct 3-dimensional structure. If that structure is in some way disrupted, then the protein very likely will not function properly in its structural role or its role as an enzyme or the many other things that proteins are essential for within cells. So protein denaturation is the disruption of the three-dimensional structure of a protein. So I'll go ahead and write that out, that denaturation of a protein is disruption of the 3D structure of a protein. And typically there, we're talking about the disruption of the tertiary structure of the protein. Why protein denaturation is important, we're going to fill in it is important. Because disruption of the 3D structure will lead to disruption of the protein function. Since the three-dimensional structure is absolutely important, absolutely essential for the function of the protein. So disruption of structure leads to loss of function of that protein. And then 3 ways to denature proteins. We'll go ahead and just make a list here. 1, 2, 3. It just says list those 3 ways. It doesn't say to extensively describe them or anything. So I'm going to do what the question asks for here in terms of what I write down on the page. And in asking 3 ways to denature proteins, one of those ways was heat. The example of that we gave in class was that if you heat an egg, eggs are loaded with protein, and the heating process denatures those proteins and causes a very noticeable change of the texture of the egg. It's going to change it from being very runny, liquidy to a very solid type of material, and that's due to the protein denaturation, the disruption of the 3-dimensional structure, leading to a very obvious change in the proteins. So heat is one way. Another way to denature proteins is with use of alcohols. In particular, ethanol, at a concentration of 70% or more generally, is very effective at denaturing the structures of proteins. That's one reason why alcohol-based sanitizers are very prevalent. Item number 3 on the list of ways to disrupt protein structures, disrupt or denature proteins, is going to be to change the pH of the environment that the protein is in. If you change the pH substantially enough, you're going to lead to irreversible damage to the protein structure. You're going to disrupt its tertiary structure. So those are going to be 3 ways to denature proteins. Coming onwards to our next question here, it says to provide the structure of the following peptide and label all the peptide bonds, R groups, the C terminus, and the N terminus. Keep in mind that the R groups are the variable groups that define each of the 20 standard amino acids. The C terminus is generally listed as the far right-hand terminal of the peptide. So it's going to be there on the L end of our sequence there. The N terminus is generally listed to the far left, and it's referred to as the amino terminus, nitrogen referring to the amine group there. So we'll go ahead and draw out this structure of this tetrapeptide. This molecule has 4 amino acid residues listed there by their 1-letter abbreviations. And remember that in this class, you will have access to the amino acid chart, so you don't need to memorize the 20 structures of the amino acids. If you ever take a full semester of biochemistry, you will very, very likely have to know those by heart, but for now, you will have your cheat sheet available to you on the exam. So in order to draw out this tetrapeptide, we consult our amino acid chart, and we're going to start by drawing out the structure of isoleucine, which you will see on the chart is abbreviated with I. So I'm going to draw out our isoleucine group in red here. It's best to orient this so that your nitrogen atom, which is generally positively charged at physiological pH, that terminal nitrogen, is on the left to get our N terminus into place here. And just draw that out as our NH3 there with our positive formal charge. And then we come over to our carbon atom. That is our carbon that will always be bonded to our R group here. And we can draw out the branch coming off of this as a line angle formula or condensed structural formula, whatever your favorite type of way of representing this is. So we'll go ahead and draw out our isoleucine group here, like so. That's the R group on isoleucine, and then we have our hydrogen that will be bonded here, and then the carboxylic acid group, which In the case of drawing a peptide, we're going to replace the carboxylic acid group with an amide linkage that leads us to the next amino acid of the chain. So we take that carboxy, which was COO minus, in our free unbonded amino acid isoleucine, I, and it is going to have formed a covalent bond, a peptide bond, or an amide linkage to the next one in the chain, which is Y for tyrosine. So in order to fill in tyrosine, we take a look at tyrosine, one of our aromatic amino acids, off of our chart here. And to fill that in, we go from our nitrogen atom of tyrosine to the carbon atom of tyrosine, which is bonded to our hydrogen and always bonded to our R group there. The R group on tyrosine is going to be CH2, followed by our aromatic ring, which has those alternating single and double bonds around the six-membered ring, and we can abbreviate that with the circle inside of the ring, and then that has a hydroxy group there at the top. We come on over, and that leads us to our carboxy side of the tyrosine residue, where we'll have our carbonyl group there. And then that carbonyl group, just like what we saw in the connection between isoleucine and tyrosine, is going to be connected to the nitrogen atom of the next residue in the chain. The next residue in the chain is proline, so I'll put P in blue there to emphasize that we're drawing the proline-derived atoms in blue. We also need to remember to go in and make sure that we fill in the required hydrogen atoms of our peptide bonds there on tyrosine to fill the octet. So what you're going to end up with here is, in order to have the complete octet, you're going to have that carbonyl group bonded directly to the nitrogen. It's going to be an NH group there in order to fill the octet with 3 covalent bonds on nitrogen and 1 lone pair of electrons, which you don't necessarily have to show, but you can if you want to. Now onward over to proline. Proline is the unique amino acid in that, if you look at the structure of it, it has a ring in which the R group is bonded over to the left to the nitrogen atom of that molecule. So we'll have to pay special attention in how we go about drawing this. I'm going to draw the skeleton first. We have a nitrogen of proline directly bonded to our carbon, hydrogen bond there, and that carbon is bonded to the R group and also bonded to the carbonyl group that will connect to the next amino acid. So now on proline, with proline we have our CH2 group directly connected, just following that around the ring, to another CH2 group, to another CH2 group, and that CH2 group is connected over to the nitrogen that is to the left of that carbon, the nitrogen that's part of the backbone. So in the case of proline here, when we look at what has to be going on at the peptide bond, since the nitrogen always needs to have 3 covalent bonds, it already has 3 covalent bonds in this case without having a hydrogen atom. In the case of the proline link of the nitrogen to the atom to its left there, the carbonyl group, carbon. And so therefore, this particular nitrogen of the proline will just have a lone pair of electrons there. It is not going to have a hydrogen directly bonded. And this is the only one of the 20 amino acids that is this way, where we will have, rather than an NH bond, which is typical there at the amide linkage, you'll have just a nitrogen because it already has its 3 covalent bonds that it needs without having a hydrogen there. Whereas in the other cases, that nitrogen needs a hydrogen covalently bonded there to get its complete octet. So this is our link to proline. And I included that in this example problem because it's a little bit of an oddball out of the different residue types, and it's perhaps the only one that you haven't really come across before. Then we go from proline to the last residue of this tetrapeptide, which I'll put in red here. It is leucine. And for leucine, we're going to connect our carbonyl group of proline to the nitrogen atom from leucine, and then looking at the structure of leucine, you'll have your nitrogen directly bonded to the carbon that has the R group bonded to it and the hydrogen, and then COO minus, because we're at the end of the peptide now. And we need to draw in our R group for leucine. R group for leucine is CH2, CH, and then there's 2 methyl groups bonded to that carbon, so I'll just put CH32 there to save a little, CH32, subscript 2 that is, to save a little bit of space on drawing leucine. So now at this point, we've drawn out the structure of the tetrapeptide, and we're asked to label all peptide bonds, all R groups, the C terminus, and the N terminus. So in labeling those peptide bonds, and making sure that we filled in all of our hydrogens that are necessary. This will fill in the hydrogen on our nitrogen there for leucine. And circling our peptide bonds in yellow to illustrate those, we have 3 peptide bonds connecting these 4 amino acids. So our peptide bonds are what I have labeled in yellow. So all 4 of these carbonyl groups directly bonded to nitrogens are what we refer to as our peptide bonds. So I'd label those out in this cartoon, that those are all our peptide bonds. You might also see those referred to as amide linkages on an exam or quiz situation. And then we're going to label the R groups as well. So I'm going to put the R groups here highlighted in blue. And for the R groups, that's what's going to differentiate each of these amino acids. So we're going to have the isoleucine group here at the far left. Tyrosine is our second one, circled in blue there. Proline is going to be this as our R group, that 5 atom ring there. And then leucine, very last here. So we're looking for what is bonded in each case to our alpha carbon, our carbon atom that has that unique R group bonded. I put the stars on those carbon atoms that have the unique R group bonded to each of them. That's what differentiates our 20 amino acids. C-terminus is going to be by convention at the far left end of the molecule. Far right end of the molecule is our C-terminus. That's also known as the carboxy terminus, where you have the free carboxylic acid group. The N-terminus is at the far left-hand end, N standing for amino. That's where you have your free nitrogen atom. That is typically NH3+ at physiological pH. So this is going to be a very typical type of problem where you're asked to draw a peptide sequence and label some features of that peptide. Now on to our question on enzymes here, and it asks what's the active site of an enzyme and what's the likely outcome if an active site residue were mutated, meaning that if one amino acid residue were mutated to some other amino acid, like a tyrosine residue was mutated to an alanine residue or a glycine residue, changing the active site of the enzyme, what's going to be the likely outcome of that? So the active site is a very important part of the enzyme because it is the portion of the three-dimensional structure of the enzyme that interacts with the substrate. So active site is where the substrate binds to the enzyme. So active site is the location of the enzyme. Where the substrate binds. The substrate has to bind to that active site in order for the reaction to be catalyzed. If the substrate binds poorly or not at all, then the reaction will not be catalyzed. So it's the location of the enzyme where the substrate binds and the reaction is catalyzed. And what very commonly happens when there's a mutation of an active site residue, that's going to interfere with the binding of the substrate. And so mutations likely lead to decreased substrate binding in most cases. So mutations decrease substrate binding. And are going to decrease the reaction capabilities, the catalysis of the reaction. And depending on the extent of the mutation, it could lead to just the reaction still occurring, but occurring not quite as efficiently, or it could lead to the reaction not occurring at all because the substrate molecule is no longer being recognized by the enzyme's active site.